Welcome to another episode of Whose Gene Is It Anyway? I am your host, as always, Justin, and welcome to a very special episode. It is going to be a fun one, I think. So, you read the title, you saw the thumbnail, you know what's up. So this one's going to be a little weird. We didn't do a vote for it, we didn't pull it out of a hat, it's just something I've wanted to work on for literally years. And I decided today was the day. We are going to do the thing. Now, what is this crazy idea? We'll get to that in a second. But before we really get going, couple of quick announcements. Thing number one, if you're not already aware, there is a new video on the main channel. So if you haven't already, I'd definitely go check that out once this is done. And other than that, you'll also know that the donation bar has changed a little bit. We're going to actually make the thing that we're going to design today, and I'm going to talk about some specifics of when that order will actually go in. I mean, thing number one, got to fill the old donation bar. But also, there's going to be a delay between when this stream is finished and when I'm actually going to put in the order. The reason being is I'm a little bit unsure of a couple things that we're going to talk about, and I will definitely get to there. But basically, because this is going to be such an involved thing, I want to make sure that we do it right. So I'm going to leave some time and basically leave sort of like a comment period sort of thing so that people can interact with the end file on GitHub once this is all done, and that way we can essentially have made the ideal thing, if that makes sense. Just because like it's expensive, so I don't want to fuck it up right at the beginning. And I actually intend on doing some testing with this, and we'll talk about all that in a minute. So if you donate through the donation bar in the description, it will, or if you donate through the donation link in the description, it'll fill the donation bar. If you donate through Super Chat, it will not, but is still very greatly appreciated. So let's kind of just get into it. I'm gonna just go ahead and quickly ban some shitheads. So yeah. Also, I have fixed the number. I realized I forgot to change the number. So now it's actually all labeled right. We already got our first donation from the amazing Nico Schluter. Thank you very much, Nico. You're fantastic, as always. Okay, cool. Quick answer to a question I see in the comments. Colin Murray asks, this isn't the main channel? No, this is the main channel. There is a new video on this channel. I say the main channel because I have two channels now, which if you didn't know that, there's a second one called The Taste Emporium, where I mostly do food. Okay, so what is this crazy bananas idea? Mr. Roberts, thank you as always. Okay, so crazy idea. I'm gonna switch this to display mode so I can show you what I'm talking about, because this one is very weird. But let's get into it. Okay, so we're set. We're ready to go. Now I just need to see. Yes, I believe this is the yes. Okay. So this is the paper that kind of started it all, and there's actually some of the main researchers actually have a company that's doing this, but the way that we're going to go about this hopefully doesn't interfere with their patent, and if it does, I don't care, because we're doing this for research, and there's no laws against doing so. Okay, so here's the thing, right? This paper is called "Targeting Human Cancer by Glycosaminoglycan Binding Malarial Protein." Let's break that down. Glycosaminoglycan binding, this is talking about these weird polymers that tend to be on the outside of cells. So I believe I have a picture pulled up. Here we go. So on kind of dangling off the ends or the outsides of cells, you have these weird thing called glycosaminoglycans. They're basically they're sort of like sugar chains. Like they're basically chains of saccharides, but they will often have very interesting functional groups. A lot of the time they've got sulfur hanging off of them in various places. They're very specific saccharides. So it's not just glucose. It's not like this isn't cellulose. Like it's very specific. They're also aminated, so they also have amino groups in and on them. So they're sort of odd. Now, why is this interesting? So let's talk about malaria. Okay, so this is the malarial life cycle. So for those who are not aware, malaria is a parasite, and it is delivered to humans usually through mosquitoes. And so basically what happens is when the mosquito bites you to feed, because they're bastards, what ends up happening is through their salivary glands, which are infected with this parasite, the parasite ends up being essentially put into your body. And they've got this very weird, interesting life cycle where they go through sort of different stages. And one of the ways that the parasite figures out what cell type it's supposed to be homing to, because it needs to get into hepatocytes. So it needs to get to your liver. So it goes to your liver, gets into the hepatocytes, which are liver cells, replicates like crazy in one form. Then it is released from the liver in this one weird form, then ends up in red blood cells where it turns into a different form, where it can then spread and then be picked back up by the mosquitoes, at which point it goes through another part of its life cycle, and then the whole thing starts all over again. It's a real bastard. So this is malaria. But part of the way that it figures out what cells it's supposed to be attacking is a collection of proteins on the surface of the parasite. And so basically, if these proteins stick to surface compounds on the outside of the cells that it's supposed to stick to, then that triggers a biochemical reaction which tells the parasite, okay, this is where I'm supposed to be, insert self into cell. What's kind of weird, though, is it has this, one of these proteins is called, let's see if I can, yeah, here it is. So one of these many proteins is called VAR2-CSA. And VAR2-CSA, I'm going to see if I can scroll down. This is the whole, I'm going to make this bigger. This is the whole protein up here. So this is the whole thing. And when this whole thing is expressed, one of the weird quirks of this, like it homes to, so I don't know if this specific one homes to hepatocytes, but this specific one has a very weird quirk of homing to human placenta. Not really sure why this ended up in this parasite, but it is the way that it is. What's very weird, though, is human placenta is very distinct from other cell types. And one of the things that makes it distinct is the glycosaminoglycans that are on the placental cells are very different than other cell types, like extremely different. Now, the weird part is there's only one other time where those specific glycosaminoglycans show up, and it seems to be in cancer cells. Something about the process of a cell becoming cancerous messes with expression of various genes that seems to translate into the expression of the same glycosaminoglycans on their outside as in placenta, which, as we just discussed, this weird-ass protein happens to stick to, and essentially nothing else. So what this paper studied was, where does this protein stick? And so I'm going to have to scroll down a little bit. This is a fantastic paper. If you want to get into the specifics of this, then I would highly recommend giving it a read. It's very dense. It took me like 3 or 4 reads. The very first time I read it, it literally took me like a day of just like staring at it and chewing on it to finally like make it make sense. But okay, so what this paper did, first let's just quickly look at the above bit. I just passed it. Shit. Okay, so they figured out that if this is the whole protein, I should just be able to reopen this. Okay, so if this is the whole protein, the bit that actually sticks to the glycosaminoglycans are actually only this very small segment of the whole protein. Like the rest of it doesn't matter. This bit sticks. This shit not so much. So they figured out, okay, well, if we just use this small fragment, we can essentially ignore the rest of it and maintain the same function. So what they then did was they made this synthetically, and then they stuck a variety of different things to it. In one case, they stuck this V5 tag. In another case, which I'll show you in just a second, they stuck something called horseradish peroxidase to it. So horseradish peroxidase is an enzyme, and we use it in a lot of different biochemical assays when we want to basically figure out when something is present. So we'll stick it usually to something like an antibody that targets a specific protein. And we're going to talk about antibodies in a second and how they work. But just for the second, understand that antibodies have little ends on them that stick to very specific small areas on specific proteins, usually. There's some variation in that, which we'll talk about. So what they do is they take the antibody against the protein that you're interested in, and they stick this horseradish peroxidase on it. And then when you expose your sample to a chemical, the horseradish peroxidase breaks it down, and you end up with this like brown pigment. So this is a really good way of seeing what the antibody stuck to, because you got to understand that antibodies are freaking tiny. Like, you're not going to see them in a microscope. You can barely see them in like an like a transmission electron microscope. They're unbelievably tiny. So this is a very convenient way of showing a truly microscopic thing on a macro scale so that we can see it. So what they've done is they've taken this horseradish peroxidase, stuck it to this weird fragment of the malarial protein, and then exposed it to a huge number of cell lines. And what they found was it essentially only sticks to cancer. And so this is lung cancer, colon cancer, rectum cancer, liver cancer, esophagus cancer, kidney cancer, pancreas cancer, breast cancer. It sticks to all of them, which is insane. Like, the fact that it does this consistently is absolutely bonkers. So then one of the other things that they did was they're like, okay, well, we know that it only sticks to cancer. Does it end up in the cancer? And yes, it totally does. Through some of the other tests that they did, I believe this is it, they found that it localizes very consistently to cancer, and also ends up inside the cancer cells. Like, something about when the protein sticks to the cell goes, oh, I'm supposed to take this inside, and it does. So this is fantastic for a lot of reasons. So then what they did was they took we're going to call this the targeting section for the time being, because it's too annoying to say it otherwise. They took the targeting section, and they combined it with what's called a cleavable linker. So basically, this is a special compound where once it's inside cells... Oh, somebody donated. Thank you very much. Greatly appreciate it. Once it's inside cells, the enzymes inside the cells cut it. And so it separates the targeting thing with whatever else you stuck on there. And so you can see it in this image here. They stuck diphtheria toxin, which is a toxin from diphtheria, which is a type of infection. I believe it's a bacteria. So what they did was they stuck diphtheria toxin, targeting thing, and cleavable linker in the middle. So what ends up happening is this homes to just the cancer cells, ends up inside the cancer cells, and then the cleavable linker is broken, the toxin is released, and then that cell dies. And this works ridiculously well, so this paper claims. This is the cleavable linker. You can see it right here. It's basically like a weird synthetic amino group thing. It's kind of weird, but whatever, it does the job. And so, they found that this works extremely well, but and also that it was very consistent at only targeting the cancer cells and leaving everything else alone. So, like, they could give a pretty reasonable dose of this weird new drug thing that they'd made, and the mice they were testing it on were fine. I mean, they'd given them cancer, so, like, how fine can you really be? But, I mean, considering that the drug, which is poison, like, they designed it to be poison, but only to a specific cell type, left the rest of the mice alone. So, this is real cool. So basically what I want to do is a slight variation of this. So rather than the cleavable linker and like having to chemically stick a drug or something to it, because this, like, you do this chemically. Like you'd make your targeting, you'd grow your targeting thing in one cell type, you'd grow your diphtheria toxin elsewhere, and then you'd have to like chemistry them together to actually make this weird new drug. Rather than that, because that's annoying, I'm a big fan of what's called immunotherapy. And so immunotherapy is basically the fairly straightforward concept that rather than treating cancer by essentially giving the patient poison and hoping the cancer dies first, immunotherapy is they're calling it the new fifth pillar of cancer therapy, where basically it's various methods, be it synthetic antibodies or modifying your immune cells or a variety of other methods, where you basically train the immune system to find the cancer on its own and just clear it, and then you don't need no surgery, nothing. It's just you do the thing and it just clears out the cancer, which, I mean, for the patient is much better because you don't get the weird side effects. You don't get, I mean, you don't need to melon ball them. Like it's a thing. So that's the idea is, okay, how can we take this, which from what we just saw sticks really well to a huge variety of cancer cell lines, taken from a variety of different patients and different tissue types and all this kind of stuff. How do we take this and turn it into an immunotherapy? So now let's talk about antibodies. This is an antibody, or a caricature of one. They're always drawn like this. They do kind of look like this, but not here. This is a better representation. So they're closer to this. They are Y-shaped, but I mean, this is even a rough approximation. But for the sake of argument here, it's this Y-shaped looking thing. All right, I'm actually going to use this picture because it looks a little nicer. So they're basically broken into 3, but really 2 segments. Okay, so we've got the targeting bit, and then the bit which the rest of the immune system recognizes. That's the easiest way to think about this. There's thing that sticks to thing that warns the immune system anything that this is stuck to is bad. That's basically how this works. So we call this an antigen. This is the thing that it'll stick to, and the antibodies are extremely specific. Although, I mean, this varies. So there's what's called monoclonal antibodies and polyclonal antibodies. Monoclonal antibodies will only, in theory, stick to one specific location on one specific protein, whereas polyclonal antibodies will stick to a variety of different proteins in a variety of different locations. And also, they don't necessarily need to target a protein. There's some weirdness where they can target other things, but for the most part, for the sake of argument here, they stick to proteins, which most organic things are basically protein anyway. So, like, this is a fairly good approximation. So what happens here's a really basic caricature. So the idea is that when the sticky bits stick to something, this will continue to happen in theory. Like this will happen many times from many antibodies, and as the load of antibodies versus thing they are stuck to builds, the immune system, so this will trigger a whole bunch of different parts of the immune system, but one of the things that it'll trigger are specific immune cells that are essentially made to recognize this. So they recognize the back end, and I'm not sure about this because immunology isn't really my field necessarily. I mean, I dabble, but it's not really my thing. I suspect there's a conformational change, which means the shape of the antibody probably changes a little bit when it sticks to something, but I can't be sure of that. I'm not sure that's correct. So the idea is the antibody sticks to the thing, the rest of the immune system recognizes that antibody has stuck to something, and then it proceeds to eat whatever the antibody stuck to and then break it down. This is how we fight viruses, this is how we fight bacteria, this is how we fight all kinds of different things. When you get infected with something, one of the ways that your body will respond is it will essentially randomly make the sticky parts until it finds one that sticks to the invader. And at that point, it'll quell the infection in theory, and then a particular cell type called memory B cells basically learn which antibody worked, and then will retain that knowledge, so to speak, so that if you ever get infected again, it can very quickly multiply and produce enormous levels of that specific antibody, so that way it can reattack the infection. It's why once you've got a virus once, you can't get it again in theory. There's exceptions to this, but like if you get the common cold, for example, you never actually get the same common cold twice. If you get it again, it's actually a different strain of the virus, and so it's different. And I realize that this whole thing is going to get demonetized, so at this point I don't even care. But the important part being that this back part, the butt end of the antibody, if you stick this to something else that sticks to something you're interested in, the immune system can still stick to this. Like, it can still target this just fine. So the idea is we're going to take the back half of an antibody and this little weird fragment of this malarial protein and shmoo them together, and then basically make it so that you could deliver this to a patient. The patient's own cells will produce this new, like, synthetic antibody thing, crank it out, that'll spread through the body, stick to the cancer cells, and then, in theory, clear out all the cancer in your body. Okay, good. We've covered the basic idea. Now, where does the HIV come in? Because you saw the title. Shit's bananas. So let's talk about what HIV is. HIV is a virus. So you saw it in the thumbnail. For those who've never actually seen what a virus looks like, this is it. This is what it looks like. They're weird looking things. So basically, HIV is a virus, right? We know this. It is actually as viruses go, it's a very small virus, and it has a bunch of very helpful properties. I say helpful in the helpful to biologists, not so helpful for the people who have it, obviously. One of the helpful properties of the virus is it's very easy to reprogram it so that it packages things other than viral DNA. So this is very helpful because you can essentially, I'm going to look this up really quick, lentiviral 3 plasmid system. So basically the way this works is the scientists who figured this out originally broke the viral genome into several different pieces, 3 in this case. Sometimes they come in 4. It really depends. Some they used to come in 2. Basically, the way this works is they've broken it into pieces. So let's look at this. For example, I think this is it's the AdGene guide. Let's look at AdGene. So we're gonna actually be getting something off of AdGene. We're gonna be using what's called a 3rd generation system because it's the most efficient and causes the least problems. So basically the way this works is they've broken this into 3 pieces, as I have said 100 times at this point. The different pieces are the envelope plasmid, the packaging plasmid, and then the thing you actually want it to carry. So originally, in like the core original virus, all of this would all be packaged as one. Whereas now they've broken it up into pieces so that it can't. You'll see these green areas here. They're called LTRs, or long telomeric repeats. Basically, these mark the start and end of viral DNA, in theory. I mean, this is what the virus thinks, but we've essentially hijacked this system. It doesn't actually check what's between these things. It just looks for these and goes, yeah, that must be my DNA, and then it packages it into the thing. So as long as these other genes are present in the same cell, like this one is cranking out the bits of the virus that actually make up the shell of the virus. This one makes the proteins that handles the packaging. So like taking the DNA, put it into virus, close it up, yeet it out of the cell. Whereas this is the thing that actually gets packaged. And so you'll notice that all of the important stuff that allows it to replicate is not present in the thing that's actually going to get packaged. So basically the way this works is you package some piece of DNA you're interested in. Oh, somebody donated. Thank you very much. You package the piece of DNA that you're interested in, and all this crap gets left behind. Once that, you then harvest the viral particles and then deliver them to a patient. And once they enter a new cell, they basically open up, but they have no mechanism of making more. So it's just a delivery mechanism. This isn't strictly necessary. You could do this just using like a special chemical and like plain DNA, but this is very effective and tends to work better than just plain DNA or plain DNA and a special chemical. So this is why we're going with it. So the idea is all we're going to do is we're going to find a plasmid that contains this chimeric repeat on either end, a promoter in the middle, and then we're going to see this blue piece, it says cDNA or RNA. We're going to replace this blue bit with the weird fusion protein that we're going to make. And so essentially what we're doing is we're taking a modified version of HIV, which has essentially been disarmed and made safe. We're loading it with a malarial protein stuck to half an antibody and delivering it to a patient so that these new weird compounds will flood a patient's system, stick to all the cancer cells, and leave everything else alone. And then the immune system can go in, clear it all out, and then, Bob's your uncle, here we go. That's the plan. That's the gist. This is the theme of the day. So again, in summary, we're taking malarial protein that for whatever reason sticks to cancer, combining it with an immune system molecule, using HIV-ish thing to deliver it, and using that to clear out all the cancer in your body. That's the plan. Now, what are we going to actually do? So you'll see that, there's the donation bar. We're going to actually make this, whatever it is that we end up making. If we don't hit the funding goal, I'll fucking pay for it myself. But this just kind of speeds things along. Whatever we end up making is going to probably be submitted to AdGene so that other researchers and other people can experiment with it. I also have a friend who is currently building an animal testing lab where he's going to be getting something like 150 mice, specifically to study a variety of things, one of which is cancer. So we're going to have the ability to directly test this in an actual cancerous system and see if it works, basically. So this is very, very cool. Just to answer a quick question here, why would the HIV cells stick to cancer and not everything else? The HIV doesn't. The HIV just delivers this to cells. It doesn't matter which. Whatever cells it infects will start producing this weird compound and then yeeting it out of the cell. And from there, that compound does the targeting and sticks to just the cancer. Everything else, doesn't matter. And the nice thing is you're not going to get... it's not like you're going to get HIV because there's no way for the virus to replicate. It is literally just a delivery mechanism, so that's perfectly safe. I say perfectly safe. This is untested. We have no idea if it's perfectly safe, but it's as safe as these things get. The malarial protein, and the thing is, at the very worst, what'll end up happening, because we're not gonna make this plasmid last very long. It's just going to last for as long as the cells do, but because it's inherently immunogenic, there's a very high chance that the cells that are producing this weird compound are also going to get cleared out of the patient's system fairly quickly. That's fine. That's a good thing. We don't really want this hanging around for all time. If it just does a big burst of the weird compound, clears out all the cancer, and then fucks off, that's a good thing. So this is the thing. A couple of questions. Why HIV specifically? Because it's particularly good at infecting cells. That's kind of it. We could also use an AAV. I just thought it'd be funnier to use HIV because the sentence using malaria and HIV to treat cancer is ridiculous. And we're doing something kind of serious today, so I kind of wanted it to be fun. But also, lentiviral. So we're gonna stop calling it HIV. It's lentiviral. lentiviral delivery is extremely effective. That's kind of the main thing. We could also use an AAV. I've used AAVs before, also very effective. Works exactly the same way. It's 3 different plasmids. One of them's got the LTRs. It's exactly the same. Very different viruses, and have very different mechanisms, but they both work pretty good. So we'll see. I mean, if I can't find a good lentiviral plasmid that has the things that I want, we'll switch to AAVs. That's fine. I don't actually care. I just want something that works. I'm not going for, the greatest DNA ever designed. I don't give a shit. I just want it to work. So that's the thing. Now, let's talk about some of the problems with this. Because, oh boy, is this... I make this sound really easy. It's not at all. I mean, it could be, but it's not. So, the... okay, here's the biggest problem, right? So this paper, where they've, used this little segment of the malarial protein, yeah, this was not synthesized. They cloned this. They took this out of, they took a blood sample from somebody with malaria and then cloned the DNA out of it. They didn't have it synthesized. That is a fucking problem. Because it means that they didn't post the sequence, which is very annoying. We just had a donation from Andrew. Thank you very much. And don't worry, I won't be testing this on myself. We're explicitly making this so that it can be tested in an animal testing lab properly and safely, and not going to just be injecting myself with stupid shit, because there's inherently a very high risk of this. We're explicitly pissing off the immune system. That can go bad 1,000 ways. So this is the sort of thing that, it's a really cool idea. I want it to be as open source as physically possible. I want it to be available for researchers, and I want to be able to test it in animals properly before it would ever go anywhere near a human. Literally the only reason I would ever use this on myself is if I'm actively dying of cancer and doctors are doing nothing. At which point, fuck it. I literally have nothing left to lose. But until that point, we're doing this properly. And also, just on not to get too heart-wrenchy here, but I had one of my you're not supposed to have a favorite family member, but I fucking did. A family member that was very close to me died of cancer, and being unable to do anything about it has kind of haunted me since. So I'd really like it if I could do something about it for others. So this is where this comes from, and this has literally been bouncing around in my head for fucking years. So that's where we're at. This is why we're doing this. It's important to me, and if I can do some amount of good for this stupid fucking planet, that would be really nice. Okay. Very cool. For fuck's sake. Okay, you're getting timed out because that shit's just beyond asinine. Okay. So, let's now talk about why this is difficult. As I said, they just cloned this out of some actual malaria, which is annoying because I don't have access to malaria, nor do I want access to malaria, nor do I have any interest in interacting with malaria. As a rule, I like to keep things that are fucking pathogens the fuck out of my lab. Because it seems important. And because I run a Biosafety 1 lab, which means no pathogens. So it just, this is the paper. If you want to look it up, this is what it's called. It's called Targeting Human Cancer by Glycosaminoglycan Binding Malarial Protein. If you just look up VAR2 CSA, it's literally one of 2 results that comes up. There's also an entire, as I mentioned at the beginning, there's literally an entire company working on dealing with this protein and making interesting things from it. One of the other papers that they wrote is this one, where they basically bound the bit of the protein that actually does the sticking is they've called VAR2, just for conciseness. It's recombinant VAR2. It's the shorter version. That's what that means. So basically what they did was they essentially stuck this targeting thing to a piece of glass, essentially. And then they can flow blood over it. They did it in a little flow cell, and so you flow blood over it like a blood sample, and it'll only stick to the cancer cells, and then you can essentially count how many cells stick to the little slide thing, and from that you can very quickly tell how much cancer is like floating around in your system. And so this is a very quick, useful test for this thing. So yeah, it's a very cool system. That's just kind of the main thing. This is just cool as hell, and I want to work with it. So yeah, as I said, they cloned this out of malaria, which is kind of a pain in the ass because that means getting it is hard. Also, it's remarkably hard to find this fucking protein in the literature. There's a lot of papers that talk about it, and say that like, oh yes, we used these variants, and it's like, okay, well that doesn't really help me much, like. So here's some important information. This paper is called "The Influence of Subunit Composition and Expression System on the Functional Antibody Response and the Development of VAR2 CSA-based Plasmid." Blah 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 blah. It's a long fucking title. Shorten your shit, guys. The important part is from this first paper. If we look at this, their little graphic here, the bits that we need are ID1, DBL2X, and ID2A. Okay, that's almost meaningless until you go to this paper where it gives you this lovely chart and tells you, so if we look at the top of the chart, it says domain borders, so it's labeling the chunks of the protein and from which amino acids in the thing that it starts and ends. So we want ID1 to ID2B, fucking, yeah, 2A. So if you scroll down, ID1 to ID2A goes from N386 to D1025. So the N and the D are amino acids. Amino acids are referred to in a variety of different ways. And one of them is what's called the one-letter code. There's also the three-letter code, and then there's the full name. So the one-letter code, let's just look this up. Amino acid letter code. So here's a chart. Go to images. All right, here we go. So if we look at this, here's the one-letter code. Here's the three-letter code. The N stands for asparagine, and the D stands for aspartate. Here's what these look like. Asparagine is right here, and aspartate is it's aspartic acid. It's this one. So you can see N, D, D's are down. So where is the thing? Okay. So I found this website, and I can't go back to the main page, but basically this website is a database of malarial proteins, so that people who study malaria and are trying to make new treatments for it have a database of information that they can draw from. So I looked up VAR2 CSA, and these two popped out. So I downloaded I clicked protein, and so it spit out this long stream of letters. You'll see that these are all individual letters. This is the one-letter amino acid code we just talked about. And in theory, this is most of VAR2 CSA. Now the trick is, which part of this is the thing that we want? So let's talk benchling. So as we've talked about many a time, I'm going to go to the website. This website's called Benchling. It's my go-to DNA editing program, which, yeah, that's a thing. Of course it is. So I actually already have this pulled up. So this is what all I did was I went from this one, copied this, and I pasted it in here. So I'm going to just open the main folder just so that we have it for in a minute. So you'll see that I've got a couple different variants in here. I grabbed both variants. So this is A and this is B. They're the same protein. They're just from different strains of the malaria. And you'll see, like, you can actually see if you look at this graphic that they are ever so slightly different. So, like, for example, right here, there's this weird colory bit here, but no weird colory bit over here. Kind of strange, but is the way that it is. And so this is just kind of like a nice visual graphic to make, caveman eyeball make sense of what is essentially just a string of letters. I downloaded both of these. So let's have a look at them. I just pasted them in. It gives you some biochemical properties, so it tells you how long it is. It gives you the molecular weight in Daltons. Normally this gives it to you in kilodaltons. I don't know why that gives you in Daltons, but I assume that's if you're making something really tiny. Daltons, it's just a mass for proteins, basically. Doesn't really matter. Gives you some other information. None of this is important. So if we remember from, where was it? This. Okay. We're looking for N number 386. So we're looking for an N in approximately position 386 in the protein. So if we scroll down, this one's the unlabeled one. Okay, let's grab it from the other one then. I labeled B. So yeah, if we look here, you can see I've already done this. So it numbers all the amino acids for you right here. So I had looked at about position 386, and sure enough, there's an N. It's right there. It's right here. And I actually compared this between both, and we'll do that in just a second. But so I grabbed that N, and then we're looking for D1025. And admittedly, I'm making this look really quick. This took me hours, both last night and this morning, just to find all of this stuff. So we're giving you the short version because that shit took way too long. Would have been boring as hell to watch. So now 1025 is here, and you'll notice there's no D. Instead, the D is ever so slightly further over, and we're going to talk about this in just a second. So the thing is, whatever version of malaria that this paper was using is not the variation that this website gave me. One of the things that was noted in, I've got a bunch of other papers pulled up, and I'll post a link to like a Google Drive or something in the description once this is all done. In all these different papers, one of the things that is mentioned a lot is that this protein is what's called hypervariable. So there's lots of little variations to this protein, so it makes sense that the version that this website carries, or this database carries, is not going to be exactly the same as this paper. So take that with a grain of salt. So now let's do something interesting. I've got these two different versions of the protein here. Let's have a look and see how they're the same and how they're different. So I've actually gone ahead and done this. I went to this little tab that's called alignments. I hit create alignment, and then I just picked the other protein. So we're going to look at this one. So what this is going to do is this is basically going to take the two proteins and kind of see where they're the same and where they're different, and then it's going to tell us that information. So for example, if we go to this end bit here, you'll see that there is huge variation just between these two proteins. So for example, this one is lots of amino acids are different, and as we scroll over, you get these dashed lines. That means this bottom one here is literally missing all of these amino acids. They're not there. So this is fascinating to me. This probably messes with some function of the protein, but luckily it's not in the area where we care about. So you'll see that I've already got this marked. So let's start at the beginning here. Through the alignment, you can actually see that even though there's some slight differences, so this amino acid is different, these two are different, most of this is the same, and the actual sequence leading up to it is the same. So we can be fairly certain that this is actually the start of the segment that we want, which is very helpful. So it's okay, that's very interesting. So now let's keep scrolling over, and you'll see that this is all perfectly the same, perfectly identical. Then we start getting into some slight variations here, and then we get to the crux of the issue, and why the letter we were looking for was not at exactly the same location. There's 4 letters missing here. By the same token, there could also be extra amino acids which could shift that point, essentially. So it is understandable to not find the amino acid at exactly where we want, and because it's so close, I think it's a reasonable estimate that we're in the right spot. Now, if we look at the end, you'll see that the actual ends are the same. So we still get this SKLD, that's the same. This one has a wildly different ending. This one has a wildly different ending. And there's, some slight variations. There's another 4 deletion here. So, like I said, this protein is extremely variable. Here, this one's also missing a chunk. This one has an extra chunk, depending on how you look at it. So, it's understandable that you're not going to find the amino acid at exactly the right spot, but it gets you to kind of the right-ish area. And the fact that these are similar enough, to me, screams this is probably right. And also the fact that this chunky bit right in the middle here is highly conserved, which means there's no changes, screams that this is probably important. And messing with this part probably breaks it. So we're going to go with one of them. I wasn't sure which one to use, and so this is actually where we talk about why I'm not going to have this synthesized like today. I'm going to wait. I mean, again, first off, we've got to raise the funds for it. But again, I'll just pay for it myself. That's fine. But one of the reasons why I'm not going to get this synthesized like tomorrow, for example, is once we make the plasmid, like the final plasmid, which we're gonna get to, I'm gonna leave it up on GitHub for a little while, because anyone who is a biologist and watching, if you can find a better version of this protein, like of this little section, and you can redesign or basically just sub this out in the file that I'm going to post at the end of all of this. If you can find one that is, if you could find the one from this paper, then that would be fantastic. If you can't, then we'll just go with whichever one of these we end up picking, and then we'll give it a try and see if it works. I'm going to add some sections to whatever plasmid we end up making to make it so that if this doesn't work, if we need to switch to a different one, it's very easy to just cut it out, stick in a new one, call it a day. I'm also going to make it so that this whole piece of DNA, like the whole targeting section and the antibody tag, can be removed in its entirety and then stuck into a different plasmid if we decide that, say, lentiviral delivery is the wrong choice. So if we want to switch to AAV delivery or a mini circle or something else, we have that option. So those are all little things that we're going to do in a minute, but we will get to there. So for the time being, I'm just going to go with this protein, this highlighted segment. We're just going to go with it because I don't have anything else to start with. I mentioned how difficult it was to find this. It was really freaking hard. Like, I'm used to being able to find genes pretty quickly. Like, UniProt does not have this. Not in its entirety anyway. Like, there's fragments of this protein floating around on the various databases, but getting the entire thing was very hard. So we're gonna go with it. The other thing I checked was, so I mean, if you just do the math, right? Like 386 to 1025 is about 660, give or take. I mean, I'm not gonna do the math right now, but the important part being that, so I'm going to just reopen this to hopefully make that screen go away and give me back my double screen. The important thing is that it's about the right length. So as long as we can see that, okay, I'm just going to close this and reopen it because this is, so one part of this site's a little bit glitchy. So that should be closed. That'll do its thing. We're going to reopen B because that's the one that I've highlighted. So yeah, we've got the highlighted thing. So if you look at it, it's about 640. In fact, fuck it, we'll do the math. So we're looking 385. Was it 385? No, 386. So 1025 minus 386 is 640, 639-ish. Whereas this one's 644. So the fact that it's almost exactly the same length And we've got the right amino acids in approximately the right positions, plus or minus about 4 or 5 aminos, says to me that this is actually the correct chunk of protein that we need. This is very good. This in theory makes our life very easy because then we have this. The other nice thing is, because this doesn't necessarily perfectly match the thing in the paper, then it could also get around their patent, because like patents generally cover 90% sequence similarity. So if this is less than 90% similar, then it might get around the patent. But that's kind of neither here nor there. I don't give a shit about commercializing this. I just want to get this into the hands of researchers and people who can test this and see if they can actually do some good. I don't care about making money from this. It's not my point. Explicitly. So, yeah. I don't know why the comments are talking about THC yeast. That has literally nothing to do with what we're talking about, but, is the way that it is. So, I'm going to just take a 3-second break just so that I can have some water, because I've been talking about 1,000,000 miles an hour. Okay, so the other nice thing with this is, because we know the aminos, the DNA sequence itself doesn't really matter. I do have the DNA sequence, that's here, but back... Oh, this is the wrong thing. This is the right thing. I grabbed the wrong thing. This is the DNA sequence. So we could actually go through and find the exact DNA, but I'm just going to reverse translate it so that it's also a little bit more different. Basically, any amount of difference that we can build into this, the less likely it is to already be locked down by some other patent, which means we can keep it open and then let whoever wants be able to play with it, experiment with it, learn from it, whatever. So be that as it may, now we need to actually find something to stick this into, because if we don't have that, then, you know. Okay, so we're going to go FC tag lenti, and we're going to see if that brings anything up. It might not. It might give me garbage, but it's just something to start with. So basically what I'm trying to do is find a plasmid that essentially already has as many of these features as we want, so that we have to do the least digging around on the internet, because that's slow and annoying. So ideally, we're looking for something that already is a lentiviral vector. It already is... there we go, see? I'm going to just hit the lentiviral button on add gene. So ideally we want the FC tag, we want it to be lentiviral, and we want it to do the thing. And, ooh, EF1A. I like EF1A. Let's have a look at this. Okay, yeah, there's the LTR. So this is lentiviral. I'm seeing the EF1A promoter, but unfortunately I'm also seeing this, which is a fluorescent protein, which is not what we want. So, you know, swing and a miss. Oh well. I'm going to just maybe grab, I'm going to just go EF1A. So EF1A is the name of a promoter. It's basically the thing that turns the gene on. I like it because it is fairly innocuous. It's not going to really cause problems. It's not viral, it's human, so it can run at fairly high levels without pissing off the immune system, which is fine and kind of what we want. We don't want the plasmid itself to piss off the immune system. We want the thing it makes to piss off the immune system, which is a slight difference. Okay, what is this? Oh, this is TED-on. Ooh, that's actually kind of interesting. I haven't seen a TED-on system in a while. There's the front. Where's the back? I don't see the back. Alright, no, wrong thing. I'd really prefer this to not have the fucking... I wonder if there's other filters that'll give me the thing that I want. It's file concentrated. No, that's not helpful. Okay, I'm gonna go with the, just to kind of narrow this down a little bit, I'm gonna go for the things that people tend to be buying, because that might narrow shit down a little bit. lentiviral backbone, iris, puromycin. I like all of those words in that order. What do we got? Okay, there's the LTR1, LTR2, poly A signal, don't know why that's there. There's the EF1A, puromycin, I don't know where the iris is. That's not helpful. But there is a multiple cloning site right here, which we can, I mean, we could use this. This is fine. I'm going to just click on this and see what it is. There might be some stuff in here, but we'll see. Okay, I'm going to just have a quick little look-see at the comments, just to see what's going on. Okay. So this is an empty backbone. That's very good. It's not super well documented. Efficient selection of stably transduced cells due to IRES-driven selectable marker. Okay. Suitable cloning sites, BAM-HI, ECAR-I, BAM-HI, not I. Okay. I like all of those words in that order. Where the hell is the BAM-HI, though? I mean, here's ECAR-I. Yeah, fuck it. Let's go with this one. Why not? Oh, I see the IRIS. Okay, I saw the note. Okay, so I'm just having a look at this. IRIS forward, IRIS reverse. I assume the IRIS is going to be right here. For those who don't know what an IRIS is, it is... What's the best way of explaining this? It's one of several ways that you can get two proteins using only one promoter. So this is the promoter. It's the EF1A promoter. This bit says turn whatever comes on, what comes after this into, like, turn this gene on. IRIS, which is for whatever fucking reason not labeled. Label your shit, people. There should be a chunk right in here where, as this is turned into RNA, there's like an extra little bit called IRES where the enzyme that actually turns the DNA code into protein can recognize both this, but also the second one. So you get 2 bits of RNA for the price of 1. It's sort of weird. I'm not a big fan, but for what this is, it can essentially be ignored. Because I mean, it's not hurting anything. And this is puromycin resistance, so you could use this. So the reason this is built this way, puromycin is an antibiotic that will kill mammalian cells. So when I say antibiotic, that's sort of it's like bespoke poison, basically. The idea being that you could take the virus that you end up growing with this, use it to infect some cells in a dish, and then give it a little bit of time, and then expose them to this antibiotic, the puromycin, and it'll kill off any cells that are not carrying this piece of DNA. And so it's a way of making stable cell lines for various research purposes, but it's not something I care about because it's not something I'm interested in, or necessary. The other thing is the LTRs here, which is, I just noticed, mislabeled. One of these should say 5'. Basically, the way these work is before this gets packaged into the virus, it literally cuts. So it'll take this ring of DNA, cut it here and there. This gets packaged. This is left behind. So it's just kind of a weird quirk of the way the thing works, but whatever. It is the way it is. Okay. So yeah, let's go with this one. I like it. We're gonna shit, I have to leave this open. Okay, so we're gonna download this as a SnapGene file, because those typically are labeled better. Okay, we're gonna go back into here. We're gonna import DNA sequence. Choose. Oh, actually, wait. Cancel. I'll just drag it in. Yay. Okay. So we're gonna drag this in. This should now be imported. I'm gonna close it. I'm now going to, the first thing I'm going to do is I'm going to immediately make a copy of this, because I am a big proponent of non-destructive edits. Never mess with your core files. It leads to sadness. Oh, there's the BAM-HI. I see it. I see you. I see you there. Okay, so we're looking for EcoRI and BAM-HI. So let's just do that. So I'm just kind of working quickly, and I will explain what I'm doing in just a second. All right, there we go. So we can turn these ones off. I'm gonna do nodding. Okay, so there's two of those. Why are there two of those? That's not good. Okay, there's that one. Where's this other one? Oh, that is poorly designed. Oh well. Okay, so we've got our plasmid here. This is our ring of DNA. These little bits of text above the DNA, and we've talked about this a bazillion times on every single one of these streams that we've done, but we're going with it. So these are what are called restriction sites. They represent enzymes. So let's just look this one up, because I don't actually think I've shown this on a stream before. But so BAM-HI, for example, is the name of an enzyme, and this is what it looks like. Of course, this is a ribbon diagram. It would normally just look like a blob of stuff. But it's a chunk of protein with a chunk of, I assume RNA or DNA or something. That this is the actual bit that recognizes a specific DNA sequence, and when it binds to that specific DNA sequence, it will actually cut it. So if we look here, if we highlight it, and so just mouse over, you'll see that there's this little like zigzag line over the DNA, and this is actually indicating how it cuts. So in this case, it would cut kind of like this between the G's, essentially. So it's and you'll notice that it doesn't cut straight down. This little zigzag is what we call a sticky end, or a sticky cut. Basically what this means is it leaves a few letters dangling off, which we can then use to stick on the thing that we actually care about. So this is very important. So we're going to use BAM-HI, and we're going to use EcoRI, because if we use NOT-I, which again, these are all just names of enzymes. If we use NOT-I, it's going to cut twice, which is kind of annoying. It wouldn't make a huge difference. If we use NOT-I and BAM-HI, for example, it's just going to cut out all this other crap. It's not a huge deal, but I don't like it. I don't like destroying restriction sites if I can avoid it. The way that we're going to do it, we're going to cut with EcoRI and BAM-HI, so it'll remove this NOT-I restriction site, but leave this one behind. So if we ever wanted to modify this later, we could. Because it basically removes one of the NOT-I sites. I say that, but there could be one in the thing we're about to stick in here, so fuck knows. Okay, so let's grab this. So I'm going to actually not grab from where it would normally cut, because we're going to... in fact, let's do it this way. Let's do this in two pieces, because right now I'm probably just going to get confused if I don't. So let's copy this, right? No. So I'm going to just right-click, copy. It's not letting me back translate. Yeah, there we go. That's what I wanted. Okay, so we're going to back translate, which means we're going to take this amino acid sequence and we're going to turn it into DNA. What's up, Nerd Rage? How you doing? Welcome to the stream. Okay, so what we're going to do is we're going to back translate. So what that means is each one of these amino acids is coded for by 3 DNA letters. So 3 DNA letters end with 1 amino acid. They're called codons. What we're going to do is we're going to highlight the sequence. We've got it highlighted. So we're going to tell it what species to back translate for. So codon usage, those 3 letters, varies between species. There can be multiple codons that all code for the same amino acid, and different organisms will prefer specific variants to others. I'm not going to get into why, just is the way that is. So we're going to go Homo sapiens, because duh, we want the humans. And we're doing this because we can still test it in mice and it'll still work reasonably well. But if we know that it works, we're not going to have the whole fucking thing resynthesized just to work in humans. So, you know. We're going to go avoid hairpins. Hairpins are basically, if the DNA becomes single-stranded, it won't, it'll stick back on itself. So if you've got a long strand of single-stranded DNA, it'll literally like stick back, because the two halves of the DNA like stick together, right? So if you've got nothing on one side, the strand itself can stick, and that's no good. So we're going to try to avoid that. I'm not going to mess with this. We're going to avoid cut sites. Let's add a cut site. So basically, I'm going to tell this to avoid not I, that one I mentioned before. So this way, it's going to try its best to not add any of these into the thing that it makes, so that way it'll preserve the usefulness of that restriction enzyme later. This is important. So let's go preview optimization. It's going to do the thing. Save as new sequence. We're going to save it in the right spot. We're going to click select. It's probably named poorly. Yeah, there it is. Codon optimized. This is it. Yay! Cool. So we now have the DNA of the thing that we want. We're going to go Control A. Come on. Control A. There we go. And now we're going to make a forward translation just to make sure that nothing has gone horribly, horribly wrong. Looks like everything is still intact. Still ends on the D. Cool. This is important. So now we're gonna make a couple little changes to this before we actually stick it into the plasmid that we have open. Oh, somebody donated. Thank you very much. Thank you, Collie. Cool. So again, before we just go sticking this in here, we need to make sure that we've made some adjustments, because you know, you always got to check these things. So we're going to use BAM-HI, right? So BAM-HI is this. Should be to there. Yeah, okay. We're going to just copy this whole thing, because I want to we need to make sure that this is there. Like when we tell them to synthesize this piece of DNA, if this isn't there, it's going to fuck up the whole assembly. So it's got to be there. So first things first, we're going to stick that right at the beginning here. Pasty paste. So now the idea is, you'll see BAM-HI, it's highlighted, yeah. They would synthesize this whole long piece of DNA, double-stranded, and then they would, the instructions that I would tell to the synthesis company is cut with BAM-HI, cut plasmid with BAM-HI, ligate together. So basically what they're going to do is they're literally, oh shit, I should have checked that for BAM-HI. That's a problem. Let's just check that really quick and make sure there isn't a bazillion frickin' BAM-HI sites. There's only one! Yay! That could have gone poorly. Okay, so I almost fucked this up, but I didn't, so that's good. Select, cool. Oh, thank you, Julius. Greatly appreciated. Thank you, everybody who's been donating. It really is greatly appreciated. I really do hope we can get this made, because I think it could actually do a lot of good, and I would really love to get some proper research done on it, and maybe ideally a little faster than some other groups who are taking their sweet fucking time. Okay, so I had a moment of panic there because I realized that when I told this to make my DNA for me, I didn't tell it to avoid putting BAM-HI sites in there. If I'd gotten unlucky, you could have ended up with a BAM-HI site somewhere in the DNA. So if you went to cut it and end up to like assemble it into the plasmid, you'd end up with cuts in the wrong spot, and that's no good. So we've got BAM-HI on that end. We've got, did I say not I, or did I say E-core-I? E-core-I. Tell me there's none. Yes, lovely. Okay. So I'm going to just mouse over E-core-I. It's going to give me the code right here, which I'm going to just quickly highlight. We're going to copy that, and then we're going to go to the end of this, and we're going to do exactly the same thing. We're just going to paste that in, and you'll see that as soon as I did that, a little number popped up here telling you where the new restriction site is. So this is very good. We now have an E-core-I site on one end. We've got a BAM-HI site on the other. Wonderful. We're actually going to put some more stuff between this EcoRI site and this D here. So I'm going to just go EcoRI site added, because remember, it is super important to annotate your stuff. Otherwise, when someone like me has to download your plasmid, it becomes extremely frustrating when I can't tell what the fuck you did or how it's organized. Okay, so we've got this. I just made this annotation so that I can it more easily stick stuff here once we get it. So we now have restriction site on one end, restriction site on the other end, but we have a problem. The problem is this is an N. N is not a start codon. In the same way that there are 3 letters that tell what each amino acid is supposed to be, there is one specific codon, and this is conserved across most, not most, but the overwhelming majority of species that I'm familiar with. There are some slight variations, but we're not going to get into it right now. There's one codon that tells every single organism that I'm aware of that to start making protein, and that is ATG. ATG always means start, and it codes for an amino acid called methionine, which, if we go create translation, is an M. And so you'll see that the M's will show up in other places. I don't happen to see one, which is kind of killing my... Oh, there's one. So M's will show up in other places in proteins, but they also always mark the start of a protein. So if we forgot to put this in here, it would literally read this whole piece of DNA and not start making protein until it hit here, which is a problem because we would be missing like 3/4 of the thing. So good, we've put this in. Very, very good. Now the last thing that we're missing is the FC tag. So we're going to just see if we can find one, and I'm not going to use the whole plasmid. I'm just going to see if I can essentially find one, and then just copy paste it, stick it into the thing, and just say fuck it, have it synthesized. Kind of makes my life easier. Okie dokie. Expresses the extracellular region. No, that's not it. C terminally fused to the FC region of human IgG1. That is exactly what we want. However, let's spin the wheel of, yep, these motherfuckers don't label their shit properly. Sigh. This is what I'm talking about. This is exactly the fuck I am talking about. When I see this big, useless fucking line here, this means that whoever submitted this plasmid is an asshole. Because this contains the bit of DNA that I want. They were just too fucking lazy to label it when they submitted it, so fuck you. Sorry, excuse my extreme frustration, but like, look at these. None of these are fucking labeled. Like, the AdGene system recognizes that there's stuff here. They just can't tell me what is the thing that is there or where is the thing that's there. It's fucking dumb. Like, if you're gonna submit something to AdGene, fucking label your stuff properly. Like, fuck's sake. Fucking 10-ply, bud. Just spare parts. Alright, let's see what we get. Nothing. Garbage, garbage, useless, garbage, garbage, useless, garbage, useless, garbage, useless, garbage, useless garbage. Okay, fuck this. Let's see if I can put a filter on here that'll get me something that sucks less. So I'm going to basically sort for things which have been purchased many times. Because the chance of that, like the more times that it's been downloaded, the less likely it is to be shit. Is kind of the way that is. Okay, so I'm not seeing anything there. That's unfortunate. Let's go more filters. Let's just downgrade. Let's turn this filter off. So basically, I'm just looking for something that's gonna have the right thing. And if this isn't here, then we're gonna just search the internet for it. But I'd really just love it if somebody would have a fucking labeled plasmid. Come on, do me a solid, would you? Fuck's sake, why did none of you bastards label any of your shit? Sorry, I'm just going to get progressively more frustrated until I find the thing that I'm looking for. Secretive signal. Ooh, secretive signal. That's actually a good point. Let me guess. It's not fucking labeled. Can confirm. Not fucking labeled. Oh, you bastards. That's annoying. That is deeply annoying. I mean, the annoying part about this one is it has everything I wanted. Like, is it lentiviral? I don't think it's lentiviral, but it's like, it's got most of the stuff that I need. Okay, we're actually, now that I think about it, I actually need to add a secretion tag. Okay, so we're going to do that in like 2 seconds. Let's just, one thing at a time. FC tag sequence. Come on, internet. By the way, while I'm bashing my face against this trying to find it, if somebody can find this and post it in the comments, that'd be great, because this is fucking annoying. Yeah, see, Nerd Rage has a fantastic point here. Only evolution gets to write DNA without labeling. Evolution was here first and will keep being here long after we're gone. The rest of us have no excuse and should label. 100% agree. Fucking label your stuff. If there is one thing that you learn from these streams as you're learning how to design DNA, label your shit. It is so frustrating when people don't do this, because now I'm like having to scrub the internet trying to find a fucking sequence for like this thing that I want, when it's like I just passed all of the things. I just can't because nobody labels any of their shit. It's frustrating. Plasmid. Let's just go FC. Oh, god damn it. No, I wanted to just go back. Let's go plasmid and see if like snap gene, because there's a ooh, I might do it. Let's see what we got. So SnapGene, it's another site. So SnapGene is actually like the expensive version of Benchling. I like Benchling because it's free and it's on the internet. Whereas SnapGene wants, I think, like $1,000 or $3,000 for their software, which looks like it was designed in fucking like Microsoft 95, and it's just the worst. That's the FC tag. Okay. C2 mouse IgG. Shit. Okay, so this is close. I'm going to save this one because this one is almost the exact thing that we want, and it's labeled. But you'll see that it says mouse IgG2A. So we want human, because if it's mouse, it'll definitely piss off the immune system all the time. Whereas if it's human, it'll only piss off the immune system some of the time, which is what we want. So let's keep looking. See what we get. Mammalian expression vectors. Let's see what we find. Yeah. When you create all life on Earth, then you can stop labeling. Also, the fact that DNA does not come with comments, it's just bad design. Whoever made this thing is just, they're slipping. It's like anytime somebody's like, the human body is perfect. I'm like, you're not a biologist, are you? Like, God's creations are so perfect. Are they though? Most of the things are held together by duct tape and zip ties and fucking inserted backwards. Like, shit's just a mess. Alright, let's try a different one. That's CMV6, FCA. Let's try this one. I write code without comments, then you're the fucking problem. Oh god, not what I wanted. Is this mouse? Mouse. Shit. Let's see if this helps. Ooh, human FC tag. What's your bet that it's labeled? Spin the wheel of people who don't label their shit. F1A. FC portion. 588. Oh, okay. Wow, this looks like it was done in MS Paint. Wow. Keep in mind, the people who submitted this are professionals. They have degrees, and they're still shit at this. Fuck's sake, what is this nonsense? I mean, yeah, it's labeled, but, like, poorly. Fused to human FC in mammalian cells. Okay. Which? Like, fuck. It's like, okay, it's the human FC tag. Which one though? I don't know. This is annoying. Why the fuck? Okay, so we're also gonna save this one because there's a good chance that it's the right thing, but I'm gonna just keep looking briefly to see if we can find something better. Okay, this one is also. Oh, Jesus, I'm gonna have to like fucking squint just to see what the fuck they put in here. Who does this? Why do you do this? Why do you hate doing shit properly? Like, fuck. Okay, well, I refuse. With this sequences for construction. Ooh, this might be helpful. What is this? I mean, this almost looks like the right thing, but full length IgG. Heavy chain. I think it's just the heavy chain. Where's that thing that had antibodies on it? No, the heavy chain is the long chunk. I just want the CH2 and CH3. So I think I'm close though. I don't know what I just stumbled onto, but it looks right-ish. Constant domains, that's not quite right. FC fusion protein. Ooh, there we go. Human interleukin 2 contains fucking. God damn it. Well, we were close. Son of a bitch. Okay, well this is this is the part where I just get progressively more frustrated because I'm getting close, but I'm not quite there yet. So this says Herceptin IgG heavy chain. This probably contains the thing that I want. I'm not going to use it because it's not labeled properly, and if I'm not 100% sure of something, I'm not going to use it because this shit's expensive, and making mistakes are expensive. So we don't make mistakes. Ooh, signal peptides, heavy chain, IgG kappa, IgG lambda, for fuck's sake, FC, tag, uniprot. Let's see if this one is helpful. Not seeing it. If anybody can just find this for me, that would really speed this thing along. Because really, once we're done this, I've got to make one quick little change, and then we're basically done. So, we'll get that done. Okay, what else we got? Okay. I'm gonna just human FC tag sequence. Come on, give me something good. I mean, the problem is there's companies that sell this. And so, like, you could buy the plasmid from, that's the whole protein. I don't want the protein. When I was doing a little bit of pre-research before I hopped onto the stream, I found a company that sold a lentiviral plasmid with the FC thing ready to go. All you got to do is cut it open. It's also $600, which, based on the cost of the synthesis alone, that would almost double the price. And so I'm like, I'm gonna not, if I can avoid it. FC of human IgG1, which is what we want. What do you suppose that this paper contains the actual fucking sequences? A bunch of useless garbage, supplementary materials, that's what I like to see. Come on, something useful. Fingers crossed here. Okay. Come on, sequence data. Nope, useless. Okay. Human IgG1 FC sequence. Anything? Uniprot. Heavy constant gamma. Constant region of immunoglobulin, heavy chains, immunoglobulins also known as antibodies, blah, 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 blah. I don't know if this is right. It's close. Fuck. You know what? When I was poking around for this, earlier, I knew this was gonna be a nightmare. I fucking knew it. Ironically, the stupid malarial protein was gonna be the easy thing. CH2, CH3. That's an interesting name. See, you can tell the person who wrote this paper is a humongous nerd, and so I'm already predisposed to liking them much more. Engineering IgG1 FC, one fragment to bind them all. I'm getting big nerd vibes from this, and I like it. Oh my god, do they actually have the fucking sequence? What a concept. Okay, we're getting close. We're real close. Okay, different their heavy chain exists. Yes, that's fine. I'm liking this so far. I mean, it looks right. It's just, it's so hard to parse this amount of information live. Which, you know, that's an argument against doing the live streams, but then I don't have people helping me. Shift around a little bit. This chair is really uncomfortable. I've moved. Oh, I'm gonna fall out of my fucking chair. Okay. Optimizing occlusion. Okay. Whoop. Wait. Shit. Did I just scroll past the supplementary? No, it's the acknowledgements. There might be a supplementary at the top, which might just have the fucking sequence. Summary, introduction. Nope. No supplementary. That's really annoying. It's close though. Yeah. Biology is reverse engineering I can't fucking words. Obfuscated code. Human FC sequence. Let's try this again. We'll get there. We will get there. There we go. Complete amino acid sequence of the FC region of human delta chain. Intact myeloma IgD. Damn it. Yeah, substandard. I get the feeling that 80% of designing genes is scrambling to trying to find the right papers. Yeah, I mean, you're not wrong. That's most of it. And I'm sorry if this is getting really boring. It's just as boring for me. I really just wish I could find the fucking thing I'm looking for. Because the thing is, I'm going to click on this, it's going to be in Vitrogen, or in Vivagen, and they're not going to share the fucking plasmid sequence, because of course they're not, because they're bastards. Be nice, though. Contents. DNA. Okay, well, that's lovely, dear. But how about... what is the thing? Yeah. Ooh, sequence. Holy crap. I think I found it. What's an RTF? I don't know. Oh. Okay, then. What the fuck? This is even worse. This is worse. This is just a string of text. I've had some guy fucking messaging me on Twitter who's just been like sending me strings of DNA letters, and I'm like, can you not? FC1, FC2, FC fusion protein expression plasmid, human IgG1. Okay. What's TDS? See if that's helpful. Plasmid features. Are these in order? Tell me there's a frickin' plasmid map. I think... Wait a second. Wait a second. Do these colors match? Green, yellow... Oh, shit. Why did that just close? Why did that just close? That's not helpful. Okay, green, yellow, gray, red. Right? Right? Green, yellow. This is a different shade of red, but I'll take it. Fucking red. I think I just found it. Okay, we're gonna just do this the stupidest way possible, which is to say probably the correct way, but whatever. We're gonna blast it, because that's that's all I've got, man. Actually, wait, let's go blast to protein. Let's protein blast this. Okay, so this is a tool called Blast, which if you do any kind of bioinformatics, this is your favorite thing. And by favorite, I mean least favorite, but also your favorite, because you use it all the fucking time. So basically what this thing is doing is I just took that string of fucking letters that Invivigen shares as their sequence, because what the fuck just happened? Oh, I hit the plug to my laptop. I'm sorry if that just... What is going on? What in the fuck is going on? I have no idea. But it's gently bricking my entire computer. So I'm going to just not kick that, because that seems bad. Okay, so basically what I just did was I took that weird bit of... Holy shit, I found it. Yeah, this is it. Holy shit, I actually fucking found it. Okay, cool. Wow, that was very frustrating. Okay, so I took this weird segment of code from Invivigen, and I submitted it to Blast. Blast is a bioinformatics tool where you give it a sequence of either DNA letters, amino acids, and then you can have it either search for matching DNA letters and figure out, like, you know, are there any things in its database that match? Or if you're... What I did was I said, okay, I'm going to give you DNA letters. Find me protein. Like, find me what protein this matches. And sure enough, and this is really important, we're seeing FC, IgG1, heavy chain, constant region. That is the thing that I've been looking for. That is exactly what I want. It's 100% cover and 100% sequence identity. This is literally one-to-one with the exact thing that we want. So we're going to say that that is successfully the thing that we want. And there's no difference between a Blast and a text search algorithm other than some of its functionality, and also it's searching a specific database. It's searching the NCBI database, which is the National Center for Biotechnology Information. It's hosted by the NIH, U.S. National Library of Medicine. It's a pretty extensive database. It's not very large. You can download the whole thing. It's like less than a terabyte. But, you know, it's there. So, cool. Finally. Fucking A. So, we're going to just paste this in now. Pasty paste. And we're also immediately going to label it, because otherwise we're going to have a very bad time. So, create annotation. We're going to go FC tag. Because remember, label your shit. Actually, no. I'm going to do better than that. Human IgG1 FC conserved region. I can spell. Wow. Nope. Okay, we're just going to fucking call it. That's descriptive enough. From Invivigen. Okay, safe. Now we're also going to immediately make a forward translation, make sure that we don't have any weird bullshit, which by the looks of it, we're good. Great, cool. So we've got our restriction enzyme on the end. One thing I want to do is I want to add a restriction site between, so I'm actually going to, oh, shit, stop, come on, dehighlight. So I'm going to just, you know what, I'll just do it this way. I'm going to go all the way up to here, shift, and I'm going to annotate this part too because I realized I didn't do that. And so this one's going to be rvar2, save. I mean, less than a terabyte is not very large. I think it's only like 60 gigs. I say less than a terabyte because I don't know how large it is, but I know it's less than a terabyte. I'm comparing this to like Sci-Hub, which is 70 terabytes. So, you know, which again, if you download all of, or not again, but in case you're not familiar with Sci-Hub, Sci-Hub is the greatest resource on Earth, literally. It is essentially all of human knowledge. It's every paper ever written that they have, which, I mean, I'd say it's 90% of all papers. Not, I mean, it's not counting stuff that's in like private databases, which good luck, but all public papers they have essentially, and compressed it's about 70 terabytes, and you can download the entire thing. Elsevier doesn't like it when you do this, but Elsevier can eat my balls. So, you know, that's a thing. Cool. So we now have our IgG. We've got our rvar2. The last thing I need to do is I need to stick a restriction site in between these two so that if at any point I need to sub out or replace or fix half of this, I don't need to, you know, have the whole thing reprinted because that shit's annoying. So now we're going to go to our restrictione sites. BSSHI. Okay, let's, first things first, before I do that, let's just see what's in the thing we're about to stick this into. Because that would be really frustrating if I use one that's already in use. So yeah, BSSHI is no good. How about small one? Small one is, there's a fucking cut site. Okay, of course there is. XBy? There's two. Where's the other one? There's this one. Where's the other one? It's afterwards. That's annoying. Okay, let's just go with the list. I'm going to just go down this list until I find one that says 0. HUC, GCT. Ooh, it's blunt. Okay, that's interesting. Hin 3. I mean, Hin 3 might work, but actually, let's just try it. And, ooh, no. Hin 3 is a hard no. Okay, so I'm just going to go through this list. This is all the restriction sites that are available in like commercial databases, and I'm basically going to see if I can find one that, oh, that's not what I wanted. That is, we want 0. We want 0 cuts, which means it's not in use. I'm also kind of, like, I've seen a few, but I'm also trying to keep an eye out for ones that I already own. Because this is actually, like, this is different than the other plasmids that we've made, where I don't care because it's not something that I'm going to use. This is I do actually want to use this one, so I want to see if I can find one that I already have. Not that it's a big deal. Restriction enzymes are usually in the range of $80 to $90, which sounds like a lot, but when you get into bio, it's nothing. Ooh, NDI! Yes! Fuck yeah, I have this one. Okay. Cool. NDI. Now let's just... That's exciting. That is really exciting. Okay, let's go back to the fragment thing, which I need to rename, but that's besides the point. Okay, NDI, please tell me there's nothing in here. Yes! Yes! I love it when shit works out like that. That is so good. That is so good. It's 6 letters. Ah, could not have asked for better. Okay, let's just paste that in. And let's make a translation. Forward. Beautiful. Beautiful, beautiful, beautiful. Okay, let's just annotate this shit really quick. Before I end up sad and forget that I put this in here. Okay, annotate. Fuck yes. That literally could not have gone better if I planned it. I love NDI. I have it. It works. Works really well. I have a lot of it. I want to change the annotation color for no reason other than I fucking feel like it. I don't like I'm gonna make it green. Green for victory. Okay, cool. What? It didn't save? God damn it. Okay. Well, I'm a professional, I swear. Save annotation. Okay. So, let's just have a quick little look-see at this. We now have... I'm going to close this, so hopefully it'll make it big enough that you can actually see it. We now have our RVAR2, which is the malarial protein. We've added the FC region from the antibody. We added a restriction site to the end. We added a restriction site to the middle. We added a restriction site to the front. Fuck yeah, boys. Okay. Wonderful. Now, the last thing that we have to do is we need to make a little change to this front part, and then we can stick this all together, and then we're basically done. And then the only thing that I need to worry about at this point after that is if we have the wrong RVAR2, and if somebody has a suggestion for a better one. If I don't get any, if I don't get any suggestions on changes once this is on GitHub, then I'm gonna basically give it a week or two, and then I'm gonna put the order in, and we're just gonna get this done. It should also give us enough time to fundraise the whole thing, because this shit's gonna be expensive. It's gonna be real expensive. Right now, this whole fragment is, if you could just unclick, please. Yeah, bases 2634. So that should be, yeah. This should be basically what I expect. It'll be about $1,000 worth of synthesis, not counting. Or that's also counting buying the base plasmid that it goes into. It counts the various fees for the synthesis company and shipping and all that kind of stuff. So it should be about $1,000. It might be a little bit more. But again, once this is made, I'm going to be making this as readily available as I physically can. It's going to go on AdGene immediately, so that way any other researcher who wants it can immediately have something to play with, and then from there we'll figure it out. But we'll one thing at a time. Okay, so the last thing I need is a mammalian secretion tag. Mammalian secretion signal sequence. There we go. That's exactly what I want. Suggested a leader peptide sequence for efficient secretion. There we go. See, this is basically the biological equivalent of like stack exchange. CD5 leader sequence. It works very well. Okay, let's try that then. CD5 leader sequence. T surface. Okay. Don't know how much of this I actually need though. 24 amino acids. Serotiny. What the fuck? Edit in pinecone. Organization. Export. Uniprot. Okay. What the fuck is this? What is this website? Cell therapies. Creative synthetic biology. Software for life. Therapeutics. That's neat. Yeah, and again, thank you everyone who's donated already. Like it really is going to go a long way to getting this done. So I just want to make sure that I've got this right, because I don't want to fuck this up. It's a PDF. I'm terrified if it's a PDF. No, it's useless. Okay, CD5 secretion tag. Okay. Linker, that's not helpful. Lord Carl's asking, which cancer? All of them. That's the cool part about this. It sticks to at least 10 different varieties and 10 different tissue types. This shit's bananas. Recombinant human protein. God damn it. Give me the fucking sequence. Thing is, Uniprot has it. It's not. I just don't know how much of it I fucking need. What the hell? Where's the actual sequence? I mean, in theory, I should be able to just grab, like, that much, and it should just work, but I don't actually know if that's accurate, so I don't want to, yeah. Yeah, I don't want to just grab a random chunk. Okay, let's do an add gene search. Oh god, add gene, and see if we can just find stuff. CD5 secretion, and see if that comes up. No. CD5 tag, maybe. So we got CD55. That's not helpful. CD59. No, that's really not helpful. Let's just go EF1A, and then I should be able to narrow this down in filters in theory. The reason it so no, it won't stick to healthy cells as far as we can tell. There'll be some paper. So okay, for those who are just tuning in, this is the name of the paper that we're basing all of this on. It's called Targeting Human Cancer by Glycosaminoglycan Binding Malarial Protein. The name of the protein is called VAR2CSA, and it sticks to like 10 different kinds of cancer because of a weird surface molecule that is a weird quirk of cancer and placenta. So, okay, now I'm going to just go... Come on, tell me it's got the right fucking... No. Expression... I mean, we definitely want mammalian expression. That's a given. Vector type... I'm going to just type in secretion. Let's see what that does. Oh, somebody donated. Thank you, Simile. Greatly appreciated. Helping to push this right along. Nope. Secreted. No. New. Not looking to study the secretion of it. Like, this is the problem. Like, they say to use this, but then they don't. Yeah. They don't actually give the goddamn sequence. Secretion. Peptide. Alien protein secretion without signal peptide removal. Okay. That sounds good. Do you have a fucking sequence? Sorry, this is boring. This is driving me nuts. This is literally the last thing we need, and then we're done. And then I can go have lunch. Ooh. Yeah, lead a sequence peptide is a sequence of 16 to 20 amino acids at the N terminus. That does okay. Okay. These all look good. Here we collected some commonly used leader peptide sequences for efficient secretion of recombinant protein expressed in mammalian cells. Fuck yes. That is exactly what I wanted. Interleukin. Oh, somebody donated. Oh, it's Third Rage. Thank you very much. Greatly appreciated. And yeah, there is still one war where nuclear, chemical, and biological weapons can be used with impunity. The war on cancer. I like that. It's well phrased. Greatly appreciated. Thank you, man. Oh, we got another donation. Thank you, Cosmic Mutant. Greatly appreciated. Okay, so these are all things... why didn't I think of insulin? Of course that would work. Okay, so we're going to go with human genes because, well, duh. I think I'm going to go interleukin 2 because that's immune-ish. That could work. OSM could work. IgG2 could work. I mean, we're using IgG, so I actually kind of like that one the most. Let's do this one. Let's go, I could also do human insulin. This one's nice and short, though. Yeah, fuck it. Let's just go with this one. We're going to go IgG2. Copy. Now we go back to Benchling and my 18 bazillion fucking open tabs. And then we paste it in. What? Oh, shit. Dicks. My bad. My bad. I gotta translate it first. What the hell? No, delete. Bye-bye. Go away. Wow, fucking... Control Z. Go away. Yay! Okay, I fixed it. Okay, reopen projects. Gotta fucking post this in. It's really annoying. Thank you, Medi. Greatly appreciated. Okay, we're gonna... This was human IgG to secretion tag. Create. This is where the fuck did I just find this? This is a really good... I'm gonna save this because this is a really good page. Novo Pro Labs for the fucking win. This is truly very, very helpful. Okay, going back. Now we get to paste it in for real this time. Oh, it's so tiny. Can I make that bigger? Yay, made it bigger. Okay, back translate, please. Yes, avoid hairpins, all this shit. Okay, fuck it. I'm just gonna just preview, do thing. GC content 60%. That's fine. Shouldn't make a difference. It should just already be saving in the right spot. It is. Wonderful. Okay, now I can copy it. Copy. Matt, what the fuck? No. Copy. Jesus. Okay, codon optimized. This should be our thing. I gotta rename this really quick because that's gonna get confusing very, very quickly. Rename rvar2. Oh, that's not typing at all. rvar2 fc fusion insert with creation tag. Because remember, labeling all of your shit is important. Otherwise, you're not going to be able to tell what anything is. Okay, so let's just do that. Do a little pasty paste. And you'll notice it starts with an ATG. You might not notice that. I noticed it. Because it always starts with a methionine. Yay, methionine! Exciting. So this is human IgG2 secretion peptide. That should do. And secretion is gonna be pink, because why not? I feel like it. Everybody likes pink. Okay, so we're done, I think. I just gotta stick this into the plasmid, and then we are ready to go. Holy shit. That is actually really exciting. The one last thing I want to do is I want to add a restriction site between this secretion peptide and the RVAR2, just to make it easier, so that way, if I ever need to, I can cut these apart very quickly. This is just something I like to do. It's not necessarily 100% necessary, but it's good practice, right? Like, it's being able to cut things apart and then put them back together easily just is very good practice, because if you, like, things go wrong, things don't work. Sometimes you gotta sub things out and cut them apart and put them back together. So being able to have the option to do so is, in my opinion, extremely important. Ooh, eagle. That's exciting. Is it 6? Wait, is not I not in here? But eagle is not I. That's exciting. Okay. I fuck with eagle. Also, it's not eagle, it's EAGI, but I call it eagle because I'm a fucking nerd. Alright, let's just do this. I should do. I'm gonna just temporarily paste that in there. Does that cause a bunch of problems? No, it doesn't. We're good. So I'm just gonna really quickly check to make sure that eagle is not in... Again, I keep calling it eagle. It's EAGI, but everyone just fucking calls it eagle. Oh shit, that's a problem. So this is actually all over the place in the other plasmid, so I'm gonna have to undo that. That's not good. Alright, well, it was worth a shot. I like it just because I have it, not for any other particular reason. Insert. This is what I want. So I'm going to undo this. That should make that go away, and then I can find a different one. That's unfortunate. I really like Eagle, because, again, I have it. But, yeah. And so the nice thing is, the way that this is designed... Oh, thank you, Will. Greatly appreciate it. The way this is designed, it's actually quite easy to test for its function in cell culture rather than skipping straight to animals. And the reason being, the FC tag is very easy to target with other antibodies. That's literally the whole point. So because of that, it would be very easy to, get a like mouse anti-human FIT-C tagged antibody, which, to put that in English for people who are not biologists, antibodies are always labeled as what species they target and what species they're from. So if you go mouse anti-human, it's a mouse antibody again that targets human antibodies. So if it's like mouse anti-human, that means it's targeting human antibodies. Whereas if it's like mouse anti-tubulin, that means it's targeting tubulin. So yeah, mouse so you get mouse anti-human, and the FIT-C is a type of fluorescent chemical that they stick onto the mouse antibody, so that way whatever the mouse antibody sticks to is fluorescent. So you could basically make this weird protein thing, fill a dish with it, like of cancer cells, and then look at those cancer cells, like stain them with the fluorescent antibody, and then look at them under a fluorescent microscope, and if they're glowing, then it works. Alright, let's see what else we got. Actually, you know what? I'm going to do this the other way. I'm going to start with the plasmid, and I'm going to see what's not here, and then go from there. Because otherwise, I'm going to end up just ping-ponging back and forth. I think I've exhausted my list of restriction enzymes which I owed. Oh, thank you, Martin. Greatly appreciate it. Yeah, I think I might have exhausted the list of antibodies I already own, so at this point we're looking at antibodies I don't own, which means I can just kind of pick whichever one I want, because it doesn't really matter. But I'm also trying to see which ones are common, because I have a vague recollection of which ones of these are going to cost an arm and a leg and a dick, versus which ones are like 50 to 90 bucks. Because like some restriction enzymes are not used very much, even though they're on this list. So if they're not used very much, then they can be very expensive. Whereas if they're used pretty often, then they're made in huge quantities, and so therefore are worth almost nothing. NSI. NSII? Okay, let's just do this thermo NSII. See what this costs. Again, this doesn't really matter. I'm just checking. Yeah, 90 bucks for 1,000 units. Okay, so it's not like it's not magical, but it's not bad either. ATG CAT. Okay, that's fine. I fuck with this. It's a bit of a weird cut, but I can make that work. Okay, so we're gonna try this one. NSII. Did I select it? Yep, it's on the list. Cool. Let's go back to our insert and just make sure that it's not already in here, because if it is, I'll be very sad. It is not. Thank fuck. Okay, so now we just grab our restriction enzyme site thing, and we just paste. That just added an NDEI site. Whatever. I'm not gonna... Wait, shit. That is actually a problem. Where did I put the other NDEI site? That's gonna cut twice. That's not good. Fuck! Damn it. Tabarnak. So annoying. Shit. Okay, well, there goes that plan. This is the like hard part. This is like when I'm actually... This is the difference between when I'm just like throwing shit together and I'm actually working. It's a very different design process. Like this, I'm actually having to be extremely careful that I don't fuck something up. Whereas normally I can be a little more gung-ho because it doesn't matter. Ooh, PM... no, it's too large. I'm being picky for a couple of things. Thing number one, I want a sticky restriction site. Thing number two, I want that restriction site to be exactly 6 letters long and not a fucking letter longer than it needs to be. Ooh, SPEI. SPEI is cheap. It's blunt though, but I don't give a fuck. Let's give it a try. It should be highlighted, and we get rid of this one. I'm just kind of working quickly now because we're almost done, and I'm very anxious to just get this done. SPI, yes, 0 cuts. That's what I like to see. Okay, wait, what? I thought this was blunt. What the fuck? Whatever, I'll take it. Okay, now we cross our fingers and frickin' hope that this doesn't introduce another weird restriction site that isn't supposed to be there. Okay, here goes. Paste to paste. Just the one. That's exactly what I like to see. Okay, we're going to annotate this. Added spy site. And we're going to make it another color because why the fuck not? Maybe bright blue. Save. Cool, that's in there. Alright, let's just do a quick little checky poo, and then we can assemble these and call it a day. We have French. I live in Quebec. I've picked up some amount of French swearing. Okie dokie. So we have our restriction site numero uno, and I just want to make sure that we don't have any. In fact, let's just pull these up. So the restriction sites that we care about are BAMHI, SPEI, NODI, NDI, ECORI. I think this one I'm not using, so this one I can get rid of. Yeah, okay, cool. There's one cut site, there's one cut site, there's one cut site, one no cut site, perfect. One cut site, wonderful. That's perfect. Okay, we've got our secretion tag with our little spacer. Then we've got our, and which one I should just make it translate. I like to make sure shit lines up. T is a little weird, but there's a T there, so it should be fine. So we've got our secretion tag, we've got our targeting sequence, we've got our spacer, we've got our piss off the immune system sequence. So that means all of this should be fine. So I click on BAMHI, and then I scroll all the way down to the bottom, and then I hold shift and I click on ECORI, and it'll actually grab at the cut sites. So now if I click copy, that should copy. All of this information should get copied when I do this. So rather than using the copy, I'm going to use the one that's named properly, just because it looks nicer. No other reason. Like literally, there's no other reason to do this way, but I feel like it. So now if I click on same restriction sites, BAMHI, hold shift, click on ECORI, and click delete, it'll get rid of all that crap. And then if I click paste, it'll put the new thing back. You'll see the BAMHI site immediately comes back. And if we click on the end here, I'm going to go all the way to the end. The ECORI site is still there. Yay! So the nice thing with this is because I have the insert... Where the fuck did it go? This. Yeah, because I have this as a separate file, this actually makes it easier to send to the synth company and be like, hey, make me this. So I want to have a quick little look at that insert. I can close this. That's the insert. I need this. I just want to see how long this is, because that'll give me an idea of price. 2692. Ooh, Jacques, thank you very much. Greatly, greatly, greatly appreciated. Yeah, so 2692, I'm just going to do a little quick calculation here. 2692 times 0.18. Okay, that's actually not bad. That's really not bad at all. So in theory, this could cost about $500 to do. So with that being the case, I set it to 1,000 because I didn't know how large this was going to be. But what are we at? We're at about 225. I'll probably adjust the goal. I think what I'll do is I'll submit this as a quote. So I'm going to put this on. Well, no, I mean, then I still need to account for the plasmid and the other stuff. So it does still bring it up to around $800. So it's not, I think it's actually going to be cheaper than I initially estimated, which is pretty good. So I'll probably drop the goal down to about 800. That should cover synthesis, that should cover buying the plasmid, it should cover all the shipping, it should cover the 10 times 100 microgram mini prep at the end, and all the services. It should cover all the restriction digests and all that shit. So it should be around $850 for the whole kit and caboodle, which would be really quite good, actually. So that's actually not bad. That's really, really not bad. Okay, so let's look at our final thing. So I can just give you a tour of the maybe it cures cancer, but we're not going to use the C word because I don't say cure until I know shit works. There's some extra stuff in here, which I don't know what it does. I don't care. I think this is... I mean, if I had to bet, I would say... Ooh, actually, I can check. I can just check. What the fuck is it? Is this the original thing? No, it isn't. I need the name of this. No, oh, come on. Ah! Cancel, cancel, cancel. Oh, not what I wanted. I just need the name of this, just so I can search it on Adgene really fast. I want to see... Because it gave an IRES thing. That'll be IRES Puro. There we go. I'm just looking at this map really quick because I suspect... Okay, the IRES starts at 3676, and go. I'm gonna close this. And goes to 450, give or take. So 3676, 450. So it's only about 100. That's not very large. That's the wrong thing. Okay. Oh, my numbering's all fucked now because I just did that. Okay, shit. I'll just do it on this one. 3676, 36367 here to about 450, which puts it, yeah, okay, that's actually right about what I expected. So if I was a betting man, which I'm not, but if I had to bet, I would say that this bit that I've highlighted right here is the IRES thing that I mentioned a long time ago. I'm gonna just annotate that really quick because the fuckers who made this didn't. The bastards that they are. So yeah, in theory this is the IRES, which only leaves a very small weird spacer between these. Ooh, I need a stop codon. But because there's an IRES here, I don't need to worry about a... what is this? It's labeled? What is the label on this? MISC feature. I don't know what the fuck that is. I'm going to assume that they didn't fuck this up and that there's a stop codon, or not a stop codon, a terminator in here somewhere, and I'm not going to bother adding one. But I've got restriction sites in case I need to. So I'm going to not add so I'm going to add, there's one change I actually need to make, and I'm going to make it in the secretion tag, and then I'm going to make it in the other thing. So it's just, I need to add a stop codon. TAA, TAG, TGA. I like to add 3. Not for any particular reason, other than I just want to make sure that it fucking works. Alright, copy that. I'm going to stick that back into the other thing, just to make sure I don't fuck this up. Is this the clean one? Yeah, it is. Okay. Oh, shit. I was already in the right spot. Fuck. Alright, that should go right there. What? God damn it. What the fuck is going on? That is glitchy as balls. Holy shit. Alright, just fucking click there, please. Alright, that's that. So, TAA, TAG, TGA. That's what I did. Enter. So I'm just, the thing I just added right here is the opposite of a start codon. It's stop codons. It's to make sure that the, whatchamacallit, fucking, the ribosome, as it's reading this bit of RNA, actually correctly, or it's from here, reads this bit of RNA, it actually stops when it gets to here. I'm not going to add a terminator right now, which is another little section that you would normally add, because I think this IRES thing means I don't need one, but also this is why I'm posting this on GitHub, and I'm not going to immediately submit a synthesis request. So, yeah, so that's exciting. I mean, it leaves me the option. So this is going to go on GitHub. I'm going to rename this right now before I forget. Hero, RVAR2, IgG1FC. Just renaming shit so that I don't forget. Oh, that's too many G's. IgG1FC, IgG2 secretion. It's not exactly the most elegant name. I'll give it a better name at some point. Something along the lines of kills all the cancer, but I'm not doing that right now. Probably make this a lot bigger before we show the fucking name. Oh no, I can go back one. I might just do it. No? No? Okay. Okie dokie. So that should actually be all of it. I'm going to submit this. Actually, you know what? I'm just going to make this. I'm going to give it a funny name because it's been a day and I feel like it. Okay, we're just going to make that copy. That should copy to the right spot. Copy. Okay. I'm just going to rename this Fuck You Cancer. I've got the appropriate name, but I just wanted one that's called Fuck You Cancer. Yeah, definitely not Zombie Virus V1. I like that one. So yeah, this I think should do the job. Thank you, Will. Greatly appreciate it. No, I'm just gonna call it no biting. No biting without a safe word. So yeah, this should be the final. This should be, or not the final version. This is going to be the version I'm going to post on GitHub. The GitHub repo, which by the way I can show you, is linked below. Almost all of the plasmids that we've made on previous streams, except the ones that I forgot to upload because I'm a turnip, and you know, shit happens. As many of the plasmids as I've remembered to post are on the GitHub. The rest will be up there shortly. This one will be up there today. Like it's gonna be the first thing I do once the stream is over. If you're a biologist, if you know what you're doing, if you have the ability to take the information which I have shared, I'm gonna post in the README. I'm gonna post as many links to the papers as I can. All of them are open access, which is nice, which means I don't need to actually like provide Sci-Hub links. But I'm going to post all of the papers. I'm going to post all of the links to all of the different things. I'm going to post the actual plasmid file itself. And that'll all be up later today. If you have the ability to double check my work, please do. And then after that, we're going to, in about a week and a half or 2 weeks is the amount of time I'm going to give it. We're going to submit this for synthesis. We're going to get it made, and it should be made in a couple of months. This way, and then from there, it'll be moving on to immediately on to animal testing, essentially. So I think this is going to be really, really cool. The fact that we might have made, and look, I'm not going to fucking toot my own horn. I'm not going to be like, I made the cure for cancer, because we don't say the C word until you have fucking data. I know a lot of people would be like, look, you cured cancer. No, fuck you, I didn't. I didn't do shit. I designed some shit on a computer. We have no idea if it's going to work. But this, in theory, could be part of the roadmap to a general treatment for a variety of types of cancer. So we're going to... so this will then be made and then sent off to my friend who's getting his animal testing lab, which is being specifically built to test cancer therapies. So this will then be tested on mice that have cancer, and we're going to see if it can actually treat them. And that's going to be amazing. And if it does, you bet your ass we're writing a paper on it and like getting it published properly, and there'll be videos. Of course there's going to be videos. But yeah, this is going to be really, really freaking cool. So thank you a million times over to the people who've donated so far. It really is greatly appreciated. I can't wait to get this order in. Again, if you have the ability to double check my work, please do. This will all be open source, like available for whoever wants it. And basically as soon as we have it synthesized, it'll be immediately submitted to AdGene. And then from there, whoever wants it can have it. I think that's basically it. The only other the only other announcements are there is a new video on this channel, which if you haven't seen it, I'd highly recommend checking it out. It is one of the better ones that I've made. It was a lot of fun. And yeah, I'm going to take probably 5 minutes just quickly to answer some questions, and then I'm going to call it for the day because I desperately need some food. I'm fucking starving. And yeah. What is for lunch? I'm having a burrito. I made homemade sour cream yesterday. because buying lactose-free sour cream is apparently fucking impossible. So I said, fuck it. I just bought some lactose-free cream, like heavy cream, and I made my own sour cream. It's gonna be delish. Can't wait to try it out. Okay, so let's see what we got. Do you have any book recommendations related to some of these topics, like a basic introduction to how genes work, etc.? The book that I always recommend is the Manga Guide to Molecular Biology. Oh shit, Jesus. Fuck. I'm just hitting wires. I'm tired. Shit's happening. Okay, this is always the book that I recommend for getting started in molecular bio, because it's adorable and also extremely well illustrated and conveys the point properly. So yeah, if you want to get started with bio, this is the book. I'd also recommend their guide to just biology. I think the organic chemistry one is still only in Japanese, which makes it harder, so then you might want to get the manga guide to Japanese while you're at it, but also just fucking... the other really great resource, which I highly recommend, is Crash Course Biology on YouTube. It's a channel that was started by the Green Brothers. Highly recommend. This is like what got me through my very first bio class. This thing's awesome. Their organic chemistry series is also fantastic. Just Crash Course, Manga Guide, those are your like best starting materials, and then from there you're getting into like the heavy technical stuff. Alright, what else we got? Are you completely working as a freelance biologist, or do you work somewhere else, or do you do this in your free time? I am a YouTuber. My job is making YouTube videos. I just happen to like doing biology, so I spend my time doing bio. I also do physics. The next video that's going to be out is actually probably going to be a COVID video, because I've been working on it since fucking December, and it's about goddamn time that I get it done. But then after that, we're going to do holograms, and that's going to be a lot of fun. But yeah, I just do this for fun, because I enjoy it, and so yes, I am an independent biologist, although I do work with, like, various academics and people around the world, and just doing cool shit. So that's me. What else do you get? How do you get as big as you are? Like, big how? Like height? Like my dick? Like what do you, like my channel? What do you, big in what sense? Okay, are you planning on working on any more bread varieties other than the beta-carotene one? Dunno, maybe. Haven't decided yet. What else we got? What about resources for synthetic biology equipment? I have made like 12 videos on my channel for everything you could need to know. How to stock a lab, how to build a lab, how to do a lot of the basic protocols, how to build a lot of the basic hardware, how to do a lot of the complicated protocols, and how to do some of the complicated hardware. I've tried to make my channel as informative as physically possible so that if you want to get started on bio stuff, you can. Thoughts on pipe cloning? I've only tried it once. I like the idea. It sounds very cool. I've just never really worked with it. If making vinegar is just oxidation of alcohol, why can't I just stick an air stone in some vodka and make white vinegar? You can. If you get... Okay, so if you're looking for fermentation stuff, the best book that I recommend is the Noma Guide to Fermentation, and I'm going to pull it up because I'm going to fucking shill this stuff every time I can. Noma Guide to Fermentation. This is the best book on fermentation I've ever fucking read. Noma is the second best restaurant in the goddamn world. Get a copy of this book. It's amazing. There's an entire section dedicated to vinegar. Yeah, so that's a thing. You totally can just run a bubbler through alcohol. Like, they made a bourbon vinegar where they basically cooked down an entire bottle of bourbon and then added a second bottle of bourbon to that, like, reduction, and then just ran an air stone through it. And also, I mean, you also need the bacteria. The bacteria go a long way to help it along, so you do have to inoculate it. A little bit of vinegar mother will make it do the thing. But yeah, technically enough oxygen will just do it. It's just really slow. The bacteria make it go a lot faster. You own the book, but it's stuck in the mailbox. Like it's stuck in the mailbox, or it's just like waiting to be delivered. What does that even mean? So for what is the bacteria, just go buy a bottle of apple cider vinegar, like something that's not pasteurized, and just add a little bit to the vinegar, or the thing you're trying to make into vinegar. The bacteria are already in there. Vinegar is a fermented product. It's a live product usually. So unless they've boiled it, it should be fine. If you buy a bottle of apple cider vinegar and it looks like it's got some like shmoo on the bottom, that's the bacteria. Just pour some of that into the vinegar you're trying to make, and it'll just do the thing. Alright, it's 7:30. I'm calling it for the day. This has been a lot of fun. Thank you everybody who's donated. Thank you everybody who helped out and hung out and all that kind of stuff. This has been a lot of fun. The files will be up. If you can, please double check them for me. And yeah, that's all. I hope you guys have a great day and the rest of the evening, and wherever you happen to be. Stay safe, stay healthy. Have a good night. Just be good. And I'll see you guys on Tuesday. Yeah, Tuesday will be the next one. See ya.